All right, hello everyone. This is going to be the start of a new unit and a new chapter. This is going to be Unit 4. We're going to be looking at mostly cell division in this chapter, but this is going to start out with Chapter 9, the cell cycle. Let's get into it. So before we talk about the cell cycle, we have to talk about chromosomes more specifically, because in the next two chapters, we're going to be talking about these chromosomes and how they split. So chromosomes are tightly packed structures of DNA and protein. Usually in my biology classes, I talk about how they're DNA just packed together, but there is a bunch of protein in these molecules or in these structures. These histones that you see up here, these are the proteins that we're talking about. And what's going to happen is the DNA is going to wrap itself around these histones to package it up. Now, the reason why we want to package the DNA is because we're going to want to move it around. When we package something up, we're not going to be able to use it. So whenever we think of DNA in this form of the chromosome, you're not going to be able to use it because it's nicely packed, away, and it's not going to be able to be read by proteins that we're going to need to read the DNA. So in other words, this only occurs during mitosis when the cell splits. Normally, we're going to see, I'm going to move forward just a little bit. Normally, we're going to see it in this chromatin form where it's nice and spread out and you can read it. But this is like making DNA nice and compact, almost like closing a book. It's good for traveling, but it's not good for reading. Okay, chromatin. This is the thread-like structure of DNA and the histones. You can see the chromatin fibers. They're going to start to condense into this chromosome form. Now, this is what a chromosome actually looks like. This is an actual photo of a chromosome. You can see those threads, those chromatin structures. Yeah, it's a chromosome. Now, there are a couple other words that we have to know here. This is where it kind of gets a little bit tough. Chromatids are the arms of the chromosome. These are specifically, this is the arm and this is the arm. You can say short arm. This is the short portion of the arm. This is the long portion of the arm. But like each one of these chromosomes has two chromatids. The centromere is the location where the chromatids are going to attach. So again, centromere is in the middle. Chromatids are what is attaching together to form the chromosome. Now, when we say that the chromatids are sister chromatids, these are going to be attached at the centromere. Like I said, these sister chromatids will have identical strands of DNA, which means, again, we're doubling the amount of DNA. We're copying it. We want the exact same DNA on either side. So these sister chromatids are going to be attached at the centromere. Non-sister chromatids, like these two structures right here, this structure and this structure, they're non-sister chromatids. Or these ones here, they're also non-sister chromatids. They do not have the same exact genetic information. They might have the same genes, but they might have different alleles. We'll come back to those terms later in the next couple chapters, but honestly, just remember that these sister chromatids have the same exact genetic information. I mean, to the T, they're exactly the same. Homologous pairs of chromosomes. These are pairs of chromosomes with identical features. We talk about these with, you get one from mom, you get one from dad, and these chromosomes will have usually the same length, the same type of genes, and they have the same features between the two. So you can see here we get one for the purple one's from mom, the bluer purplish one is from dad. I relate these to like having like a left and right shoe. So everyone has a left and right shoe in a class, but only one left shoe will go with one right shoe, if that makes sense. Like a pair of shoes will go together. Your left shoe will not go with somebody else's right shoe, so it's specific to the shoes. And that's just an analogy, but these are homologous pairs of chromosomes. And in our bodies, we usually have 23 pairs of these chromosomes, which means we have 46 chromosomes in our cells. All right, diploid versus haploid. Diploid is 2N, haploid is N. This is just denoting the difference between the different cell types. Your somatic cells or your body cells will have the average number. So our cells will have 46. This includes both kinds of each chromosome. So you can see here they have the homologous pairs of chromosomes. The haploid cells will only have one of each of the chromosomes. This is because, again, these are like the sperm and the egg. This is why you only get 50% of your genes from your mom, 50% of your genes from your dad, is because you only get one of each homologous pair of chromosomes from either. So 50% of your chromosomes will be from mom, 50% will be from dad. All right, this is the end of 9.1. Now, I usually don't show videos when I'm doing my screencasting here, but I'm going to show you, this is a frog embryo starting to split here. And you can see that obviously these cells are splitting and splitting and splitting. And the question I always raise is, why does a cell not just get bigger and bigger? Like, why does a cell have to split? So the main reason is because of something called the surface area to volume ratio. Now, there's a lot of wording here, but basically what this means is that as a cell or anything increases in size, the surface area and volume will both increase. However, the volume will increase much faster than the surface area will. So as we're increasing, and this is volume and this is surface area, the volume is going to increase much quicker than the surface area. So what happens is as a cell grows in size, the surface area to volume ratio will go down. If a cell divides, it will drastically increase the surface area to volume ratio. So again, we're comparing surface area and volume. We're comparing those two numbers. So as a cell gets bigger, the surface area won't be able to keep up with the volume. What that means, like who cares? What that means, I'll show you here. So again, we can grow in size or we can keep dividing. You can see here we have the same total volume. So if you add up all the volume of this one cube versus all of these cubes, they're going to have the same volume. However, if you look at the surface area, add up all the surface areas of these small cells, you're going to see that there is a much higher surface area compared with this one cell. So if we compare these two ratios, or we compare these two numbers and make a ratio, you can see that the surface area volume ratio is much higher here in the smaller cells than the larger cells. Why that matters is, again, the efficiency of the cell. When there is a higher surface area to volume ratio, the cell will be able to more efficiently get things into and out of the cell. What that does is it makes it easier and much more efficient at just living. These cells need to be efficient at getting things into and out of the cell, whether it's getting food into the cell or getting waste products out of the cell. If it doesn't do that efficiently enough, it could die. So we want these cells to remain small in order for that efficiency to really help with the transport of things into and out of the cell. Now, there are a couple different ways you can, not you, but cells can do this. They can either divide, obviously we're going to talk about that, but they can also change shape. So becoming long and cylindrical here, this will help the cell keep that surface area to volume ratio down. Also increasing the folds of the inner membrane. If you remember, we talked about the cristae. That will help with the surface area to volume ratio. One of the key concepts I talk about, like a couple key words in this AP biology class. Structure determines function. The efficiency is another one. Compartmentalization. This is your next one. Surface area to volume ratios. There is so many instances where we look at surface area to volume ratios. It is just mind-boggling how this ratio affects so many things in life. So this is another major key concept that you have to know of. All right, we're going to look at 9.3 right now. 9.3, this is going to specifically look at the cell cycle, and this is going to encapsulate both interphase and the M phase. So again, the cell cycle is the set of stages that take place when a cell divides. This whole thing here is showing us the cell cycle. Now, interphase, the phase here in purple, this is when a cell is, carrying out normal functions. This takes place in the cell's, like majority of the cell's life, and it consists of three main parts: G1, S, and G2. So you can see it goes through G1 first, then it goes through S, and then it goes through G2 at last. Now, when we're talking about the G1 phase, here we go. So we're in G1. Again, this stands for G1, actually stands for growth or gap phase. It's more specifically gap, but if you remember growth, that's fine with me. But this is, again, it's recovering from the previous division. It's going to grow in size. It's going to increase the number of organelles that the cell has. So again, the cell just split from mitosis from its previous division. It's just going to go and carry out normal cellular functions. It's really important to remember that chromosomes have not condensed yet. A lot of people think that the chromosomes are condensed throughout the majority of a cell's life, and they're not. In this phase, they're not condensed. During S phase, this is the synthesis phase. So think about synthesizing something, we're making something. This is when replication occurs. Replication is the copying of DNA, and we are going to see the DNA be copied. Again, we're going to get an exact copy of it. This is because the chromosomes need to split during mitosis and have the same exact genetic information. Again, remember, chromosomes are not condensed yet. It's uncondensed. So a lot of images will show the replication like this. You can see the chromatids here. They're all condensed, but that's not actually what it looks like. This is what it looks like, where we have the nice stringy spaghetti uncondensed chromosomes. This is what it more looks like, but this is really tough for us to understand. So we show it like this, but it's not actually this. It is this. I show that video. G2 phase, gap 2 phase. This is when the cell starts synthesizing proteins that are going to assist in cell division. Example of microtubules. We're going to get to those in a little bit. But again, the chromosomes have not condensed yet. The chromosomes only condense during M phase or mitosis. So you can see what that looks like. Again, the chromosomes have not condensed yet. This is what it looks like. Okay, so going back here, in this G1 phase, we have a separate kind of step or a stage or whatever you want to call it. This is called the lazy phase. But basically, these cells will not enter SG2 or the M phase. They will remain in G1 phase. It's actually called cell arrest. So what this does is it allows the cells to be more specialized, I should say. Nerve cells, muscle cells are examples of these types of cells that are not going to go through mitosis. If you think about it, whenever you have nerve damage, you don't get those cells or that function back because those nerves can't repair themselves because they are resting in this phase. So again, they're staying in this phase. It's called the GO or the G naught phase. I don't know if I mentioned that, but the cells in this phase will not leave the G1 phase. Like this meme, yeah. Cells in G1 phase or a GO phase or G naught phase, yeah, they're not going to leave that G1 phase. All right, that's the end of 9. All right, you're going to hate me, but I just added a different section. We're going to talk about mitotic proteins in this kind of in-between section, so it's 9.35. This is called the centrosome. The centrosome is composed of 2 centrioles and other proteins. This is going to be an organelle that's going to aid in cell division, and we're going to see these in right next to the nucleus. This whole structure is known as the centrosome. So we can see we have these centrioles. We have these things called microtubules. Let's learn about those next. Oh, by the way, these are only found in animal cells. Centrioles, these are barrel-shaped organelles that are found within the centrosome. They are going to organize the mitotic spindles or the microtubules, whatever you want to call them. But these structures are at almost like a right angle to each other. But yeah, these structures are within the centrosome, and they just organize these spindle fibers that we're going to see in the next section. Spindle fibers, these are also known as microtubules, like I just said. They are responsible for moving the chromosomes around the cell. You can see here, these are microtubules. They are going to be what moves the chromosomes. They're actually going to attach the the kinetochores, which are around the centrosomes of the chromosomes. There's so many words here. But these spindle fibers are going to move things around during cell division. So these 3 proteins are going to play an important role within cell division. I wanted to put them before, so when we talk about mitosis, we can talk about their function. Okay, this is 9.35. All right, chapter 9.4 is going to look at M phase. And specifically within M phase, we have 2 parts. We have mitosis and cytokinesis. Mitosis is when the nucleus divides. This is when the DNA is going to divide. Cytokinesis is when the cytoplasm is going to divide. Both of these are a part of M phase, but M phase is really named after mitosis, which is the important part here. So again, mitosis, nucleus divides, cytokinesis, this is when the cytoplasm divides. And yes, this is when we finally condense the chromosome. So up until this point, chromosomes are not condensed. This is when we finally condense the chromosomes. So I'm going to go over each part of the cell or of mitosis and what happens in it and what the cell looks like. So you can see here, prophase, first phase. Chromatin have condensed. You can see the chromatin start to condense. The chromosomes start to condense. The chromosomes consist of duplicated chromatids. There's no orientation here, which means they're just kind of randomly within the nucleus, and that nucleus does start to break down. Now, it says there, you can see it says early and late prophase. There are early, late prophase, early, late, like there's a lot more that goes on here, but I just want you to know the major steps, which are prophase, prometaphase, metaphase, anaphase, and telophase. Prometaphase, kinetochores appears. These are protein regions around the centrosomes or the chromosomes. What happens is the kinetochores are where the spindle fibers are going to attach. You can see here spindle fibers, otherwise known as microtubules. This is where they attach. The chromosomes are still not oriented. They're just kind of randomly within the cell. Kinetochores, just like I said, the regions around the centromeres where the spindle fibers are going to attach here. All right, here's the kinetochores. You can see they're interacting with those microtubules. They're what's going to pull the chromosomes apart. Metaphase, the centromeres of the chromosomes are going to align along the middle of the metaphase plate. You can see here, this is the metaphase plate. Chromosomes are now oriented in this fashion. So now they become oriented inside the cell. During metaphase, you can see that line. You can see the chromosomes here with the chromatids. You can see the microtubules here. They're going to pull those chromosomes apart. Let's watch a quick video.
This visualization DNA molecule is
So you
packaged
can
up
see here,
into
each
one of
one
the
of these
sausage-shaped
chromosomes
chromatids.
has a kinetic core
Picking
here that
out
the
the
spindle fibers
mitoside
are
are
attached to.
microtubule
All
fibers
right,
from
I'm
the
going to kind of
mitotic
move
stem.
this forward just a little bit.
This, along with the
What's
chromatids
going to
split
happen
apart
is these
to the
chromatids are
opposite
going to
poles of the
split
cell.
apart, and they're going to travel to different poles of the cell. So you can see those chromosomes and chromatids splitting apart. This occurs during anaphase. So anaphase, like I said, the chromatids split apart. The spindle fibers are actually going to shorten to do this, and the chromosomes will again be located at the opposite ends of the cell. This is also the shortest phase. Last year, telophase, spindle fibers start to disassemble. The mitosis is coming to a completion here, and the nuclear envelopes in the both of the new cells are going to start to form. Chromosomes start to unwind, and then during cytokinesis, this is the division of the cytoplasm. When cytokinesis doesn't occur, you get multinucleated cells. We'll talk about that later on and why we might see that. But yeah, cytokinesis is when the cytoplasm divides. All right, 9.5 is going to look at something called cell cycle regulation. Within this, the regulation, we have these things called growth factors. And these growth factors are ligands, if you remember from back in our cell communication chapter, that binds with the plasma membrane or proteins within the membrane. And this is going to create some internal mechanism, the signal transduction pathway, to initiate the cell cycle. So what this does is it allows your body to regulate what cells go through mitosis, because we don't want all of your cells to go through mitosis at the same time, because again, you want only certain types of cells growing or dividing. You want every single cell going through mitosis. It does this by using something called cyclins. Cyclins are internal signaling proteins that increase or decrease the amount of cellular division that occurs. And there are four main types. We're not going to really kind of dive into each main type here. I'm just going to show you that we have different cyclins that occur at different times during the cell cycle. Each cyclin has a different, initiation thing that it does to either activate or deactivate mitosis. These cyclins are going to bind with something called CDKs. CDKs are cyclin-dependent kinases. And if we think about this word, cyclin, so they need cycling, dependent, so they need the cyclin, kinases. These are enzymes that, again, phosphorylate something. These will, when bound to the cyclins, they will initiate those other proteins to work correctly. So again, this is just one way that your body is going to use proteins to either start or stop the mitotic process. So you can see here our CDKs, when there's no cyclin, it is not activated. However, when there is cyclin found within the cell cycle, it's going to bond to the CDKs, which again are going to phosphorylate different proteins, and this begins the different things that occur at each cycle. I'm not going to go over each one of these individually, but just understand the difference between CDKs and cyclins. All right, let's talk about 9.6, which is about checkpoints. Checkpoints are like locations where whatever is going on, something is checking to make sure everything is good to allow whatever it is to pass on. And in your cells, you have several checkpoints. You have a G1, G2, and M checkpoint. And these checkpoints are going to be within the cell cycle, and they're going to be when the cell decides whether or not the cell is going through mitosis or interphase correctly, and it will allow it to pass or not. Let me get into the kind of the details. This will make more sense. So in the G1 checkpoint, the cell is going to make sure that there is enough room for the cell to divide, and it's also going to make sure that the organelles have replicated. If these two things haven't occurred, then it won't go through the S phase, because there could be some errors or mistakes that occur here in that S phase then. In the G2 checkpoint, the cell is going to check to make sure that replication has occurred correctly. It's going to make sure that there's no DNA damage. And if there is DNA damage, it's going to check to make sure, and it's going to try to fix that DNA damage. Because again, if the cell hasn't gone through replication or if there's damage, then the new cell is not going to have the right amount or the right DNA to survive. So it needs to either fix the DNA or go through something called apoptosis. The checkpoint is the mitotic checkpoint. This is going to make sure the chromosomes have attached correctly to the spindle fibers and then split properly there afterwards. That is important because every new cell needs the right number of chromosomes and DNA. So if it doesn't, it's going to stop and make sure that it does have that right genetic code. Now, within the P53, within these checkpoints is a protein called P53. This is when the checkpoint is going to use a protein called protein 53. This stops cell division if the DNA is damaged. This P53 will try to fix the DNA at first. If it can't, it will stop the cell cycle and fix it further. If it can't again fix the DNA from that point on, it will actually initiate something called apoptosis. So this checkpoint specifically will stop the whole cell cycle if there's enough DNA that's damaged. Now, at that point, the cell will go through something called apoptosis if it can't fix the DNA damage. Apoptosis is programmed cell death, where the cell has instructions then to die because the thought there is it's better for a few cells to die than for some cells to become cancerous. So, this is just one of the ways that your body has evolved checkpoints to make sure your body doesn't get cancer. Now, interestingly enough, when these P53 proteins are mutated, this is actually a large type of cancer, a large grouping of cancer, because if this checkpoint isn't working correctly, then you're going to have a higher chance of developing cancer. So this is one of the more common, but it is one of the more common types of cancers when this gene is affected or this protein is mutated. I like to start out this last section about cancer with this graph. And we can see in this graph in the early 1900s, we saw a drastic increase in the cigarette consumption. And then about 20 years later, we see that same drastic increase in the lung cancer of men. And we see this because, again, it takes a while for these carcinogens to cause cancer in people. It's not like you instantly develop this cancer, but we're going to talk about why we see these trends in cancer. Here's another graph I like to talk about. You can see as we age, we have increased risk of developing cancer because we are exposed to more of these carcinogens throughout the years. So yeah, we see a larger increase in these cancers later on in life. All right, so what is cancer? Cancer is uncontrolled cell growth. This is usually caused by mutations to genes that control cell division. There are two main types of these mutations. They can be either like genetic mutations that you inherit from your parents, or they can be through carcinogens. And it's not like if you get one mutation, you're instantly going to develop cancer. You have to have usually several rounds of mutations occur. So you can see the damage that is done here. It's a lot of damage done in the karyotype. You can see the average karyotype, what it looks like, and then a cancerous karyotype. Now, this is very specific to one type of cancer, but I like to show that karyotype anyways. Now, there are two main types of mutations that can occur. You can have proto-oncogenes. These are genes that code for proteins that promote the cell cycle. Whenever they are mutated, they act like cancerous genes. So what that means is usually you have these genes that will produce they will initiate the cell cycle. Whenever they're mutated, it would be like if a gas pedal was always on in a car. This is going to cause the car to crash. You can see into this wall called cancer. On the other hand, we have these tumor suppressor genes. They are genes that will slow the cell cycle and will stop mitosis from occurring. It would, if there are mutations on these genes, then the cell would not be able to stop mitosis and it would continue to go through mitosis and maybe become cancerous. These would be like the brakes of a car. And if they didn't work, you can see here the brakes don't work. Yeah, the car is going to hit into this wall because the brakes aren't stopping the car from going. Now, there are a lot of different types of carcinogens out there. I'm going to go back. Carcinogens are just cancer-causing substances that can cause to these mutations. Radiation is obviously a type of carcinogen. Smoking, chewing, unhealthy eating and diet and drinking, environmental. There are a lot of different carcinogens. Now, it's not saying like if whatever, you expose yourself to one of these carcinogens, you're instantly going to develop cancer. It just increases your risk of developing cancer. A tumor, this is a cancerous mass or cancerous mass of cells. A benign tumor is usually defined as a cancer that's not spreading. A malignant tumor is usually one the cells are starting to spread.
And that process is known as metastasis. We're going to go over that here in a little bit. Go back. Cancer cells lack differentiation and will eventually lead to loss of the function of the organ. So the reason why cancer is so deadly is because whatever organ that cancer is in, that those cancer cells will essentially stop that function of the organ. So if the function of the organ isn't working, you're also going to have some major problems through that loss of function. It says cancer cells have abnormal numbers of nuclei or chromosomes. This is because, again, they're going through mitosis very rapidly before cytokinesis could occur. Like I said before, metastasis, this is just when the cancer cells are moving throughout the body through the circulatory system. And this is very hard for a person to fight because, again, now you're fighting on several or within several uh, like organs, so it's tougher for your body to fight. Then angiogenesis, this is the formation of blood cells to help the tumor survive, and this is really just crazy how it works. Your cancer cells are 99.9% like your other cells, and your body can't tell the difference. So because of that, these cancer cells will actually trick your blood system into getting more blood supply to them. So it almost like tricks your blood system into giving them more nutrients to grow. Um, you can see the cancer is number two. It's number two killer. Just because, again, there's a lot of people that are growing older, so there's more cancer in those people, and they're dying of it. I've seen some like misconceptions about how there's been more and more cancer. No, we're just getting better at identifying what is cancer. I like to kind of just breeze through this last part. These are some types of treatments that have been done in the past, and they're not. I'm saying they're not used; they're still used today. But we're trying to get away from these. Chemotherapy treatment that uses powerful chemicals to kill fast-growing cells. Again, this is very hard on the body. So is surgery when you remove the tumor. Radiation, you're killing the cells through radiation. So there's a lot that's going on that's really tough on the body, but it's what we used in the past. Today, we're using more things like immunotherapy, where we're going to use your immune system and you get medications to initiate these antibodies to attack your cancer cells, and it's much lower side effects and just much more success because it is more directed at the individual person's cancer. So, like I said before, total cancer deaths have been increasing because we've been doing a better job at identifying cancer, but the the rate of death has drastically decreased because we're getting better at treating cancer. So, this is going to be the end of chapter 9.